Welcome back to another video in this module where we are discussing the biology of MHC and antigen presentation. We're going to continue our discussion of antigen processing, now looking at the processing of exogenous antigens, those antigens that are derived from outside of the cell. So we have another figure here where it's a little bit of an overview of the various steps of exogenous antigen processing, and then we'll sort of zoom in on each of them one by one. But notice that we start with some sort of protein derived extracellularly. So this is something that is derived from the extracellular space in some way or another. We'll see that there are multiple ways to get sort of access to an extracellular protein, but it's going to enter the cell generally through something like endocytosis or phagocytosis, and rather than being degraded by the proteasome, which is what we saw for endogenous peptides or endogenous antigens, extracellular proteins that are going to serve as antigens are degraded in the endocytic system within endosomal compartments. And that relies on the acidic environment of the endosome as well as acid-sensitive protease enzymes in those compartments. And so they are degraded there into smaller peptide fragments, which are compatible for loading onto MHC class II. And you can notice that is actually within the endocytic compartments that they encounter MHC class II, which itself starts its life in the ER just like MHC class I, and you can notice that it is associated with a chaperone molecule known as the invariant chain. And there is an abbreviation for invariant chain, which is a little bit confusing. It's capital I, lowercase I. And in a font like this, that's a little bit ambiguous looking, but that is indeed what the abbreviation is. But the invariant chain, it both stabilizes the MHC class II molecule and also prevents premature peptide loading. It blocks the peptide binding cleft. And it is not until the MHC class II actually leaves the ER and fuses with an endosome containing potential peptide antigens that the invariant chain can be displaced. Antigen can be loaded into the antigen binding cleft, and from there, continuing through the endocytic pathway, MHC class II is targeted to the cell surface where it can display its antigen. And in this case, because it's MHC class II, it's going to present its antigen to a T cell expressing the co-receptor CD4, which is part of that overall interaction with the TCR complex. So this is a sort of summary schematic of the different steps that we'll now discuss in more detail, as I said, one by one, starting with what is the source of extracellular protein that we might use to process and derive exogenous antigens. Well, there are two sort of major sources that I think are worth knowing, and I think cover most of the examples that we would encounter in nature. On the left side, you can see that actually one source of endogenous antigens is actually pulled in through endocytosis, and this is exclusive to B cells. When BCRs bind to their antigens, recall that BCR activation requires cross-linking, so you need some kind of polyvalent antigen that multiple BCRs can bind to simultaneously. When that occurs, we saw many downstream activation events that occurred. That was in another video. But one of the things that occurs in addition to the kinase activity and transcriptional activation and all of that we saw before is also the induction of endocytosis. So the cross-linked B cell receptors will trigger endocytosis and be pulled into the cell, into an endosome. And it's not only that the BCRs will be pulled in, but whatever antigen it is that they're bound to will come along with them. And within the environment of the endosome, endosomes progressively acidify as they mature. And the combination of that acidic environment, as well as a set of proteases within the endosome that sort of turn on in response to increasingly acidic environments, these proteases also tend to cleave those antigens that have been pulled into the endosome and degrade them into smaller fragments, which is what you're seeing here in this image. And those smaller fragments then can theoretically be used as exogenous antigens. In addition to the proteases that the endosomes contain naturally, endosomes can fuse with lysosomes to create structures called endolysosomes. And the proteases and other enzymes in lysosomes sort of further the project of degrading exogenous proteins into exogenous antigen peptides. On the other hand, in other types of cells, other professional antigen presenting cells, which remember were contained within a family known as mononuclear phagocytes, so this would include monocytes, macrophages, and dendritic cells, they primarily internalize their antigen through the activity of phagocytosis. And so, using a variety of phagocytic receptors like FC receptors or lectins, um, when they bind to pathogens or pathogenic material, they pull in that material into a phagosome structure, which is analogous to the endosome. It just sort of originates in a different way. And similar to the endosome, it also is going to progressively acidify and activate proteases, which are going to degrade the pathogen or whatever pathogenic proteins were pulled in. Similarly, the phagosome can also fuse with a lysosome to form a structure called a phagolysosome, which will be even more inhospitable to those pathogens and to their proteins and will lead to further degradation. So you can see here that there's multiple ways to sort of accomplish the same thing, and they do tend to be somewhat cell type dependent. B cells do not phagocytose anything. The only way that B cells pull in exogenous antigens is through BCR cross-linking dependent endocytosis. Whereas the other professional antigen presenting cells, they heavily rely on phagocytosis. They can also endocytose, and they can also sometimes pull in antigens through things like macropinocytosis, although I would say phagocytosis is probably the most important and the most common way that they internalize antigens that they then want to present to MHC class II. So however we derive these antigens, their main processing steps are within these endocytic or phagosomal structures, but after they are degraded, we need to get them onto the MHC class II molecule. So let's look at how that occurs. So there are a few molecules here that we want to introduce ourselves to. I said previously that MHC class II also is generated in the rough ER. It needs to be translated off the ribosome, and it needs to be folded. A primary chaperone molecule that we should discuss here, though, again, as I said early on, is the invariant chain. We get the idea that it is invariant because there is very little sequence diversity associated with the invariant chain. It can bind to the peptide binding groove of MHC class II, but it itself is not an antigen. It just sort of holds space. For that reason, we think of it as invariant because it's not antigenic. It is not variable in the way that antigens tend to be. It's a placeholder, and it's going to keep itself in the peptide binding groove. And again, that serves a chaperone function, but it also prevents premature loading of antigens into MHC class II. Remember that the endoplasmic reticular lumen is full of endogenous antigens that have been shuttled in through TAP, as we saw in the last video. And so some of those antigens would be of a size that would be compatible with MHC class II binding. But because the invariant chain blocks the peptide binding groove, that excludes those endogenous antigens from being able to be loaded onto MHC class II, which we want to reserve for exogenous antigens, which are elsewhere at this point. So MHC class II buds off of the rough ER with the invariant chain in tow. And as it buds off and enters the endocytic system, just as we've seen before, the endosome is going to progressively acidify and sort of activate protease activity. And a combination of those activities are going to degrade the invariant chain. There are several intermediates of the invariant chain, but sort of the final and most important remainder of the invariant chain is a molecule called CLIP. And CLIP is the part of the invariant chain that is sort of the last to hang on, and it is the part that sits right in the peptide binding groove. So it continues both to stabilize the MHC class II molecule as well as to block the binding groove from binding anything else until the time is right and it's going to encounter the exogenous antigens in a compartment that contains such antigens. And this endosomal compartment that has the MHC CLIP complex, we eventually begin to call it the MHC class II loading compartment, or M2C. And when the M2C fuses with an endolysosome or a phagolysosome, one of these compartments that is full of digested exogenous proteins and therefore has many short peptides that can be loaded in MHC class II, CLIP is then free to dislodge and open up the peptide binding groove, and antigen is swapped in its place. And that antigen will serve the function of stabilizing the MHC class II alpha and beta chains to make sure they stay together, but also, of course, to serve as an antigen to be presented on the cell surface. So this is, or these are the major steps in this process that I think are worth knowing, although there's just a little bit more information that we should go over in terms of how exogenous antigens are successfully loaded onto MHC class II. So there are a couple of related molecules associated with MHC class II known as HLA-DM and HLA-DO. And you can notice from the way that I've drawn them that they also have this double chain structure, and they look a lot like MHC class II. That's because they are related. They're structurally and as well as kind of genetically related. They're similar molecules. But HLA-DM and DO, they do not bind antigen directly, but they serve as chaperones that facilitate antigen loading onto MHC class II. So they have a role to play, but it's not directly interacting with antigen. Instead, the chaperone HLA-DM, it sort of comes along for the ride into the M2C, into the MHC class II loading loading complex, the vesicle that contains MHC class II CLIP, and so it's present there. So when MHC class II encounters the exogenous antigens that it might be able to use, HLA-DM actually stabilizes the MHC class II alpha and beta chains long enough for the swap to occur, for CLIP to come out and for the antigenic peptide to go in. Without the help of HLA-DM, basically, as soon as CLIP left, there would not really be enough time for the antigen to come in, because almost immediately the protein would lose stability. So HLA-DM chaperones it so that there's enough window of time after CLIP is displaced, the antigen can come in and bind to the peptide binding groove. And recent research suggests that HLA-DM also has other functions. It also probably has a kind of editing function in sort of facilitating only the most high affinity peptides to load into MHC class II as well. Although by far the most widely established function of HLA-DM is more about the exchange of CLIP for whatever peptides happen to be around. So as you can see, that is a function that is supportive. It is pro-antigen presentation. It's going to facilitate the presentation of exogenous antigens on the surface of the cell eventually. In contrast, HLA-DO is a negative regulator of this process, and it does so by inhibiting HLA-DM and preventing its association with the MHC class II CLIP complex. So as you can see here, HLA-DO is kind of sequestering HLA-DM and blocking it from binding to its other potential partner, the MHC class II molecule. So in the presence of HLA-DO, if HLA-DM is inactive, that basically means CLIP will never be displaced, and the exogenous antigenic peptides will not have any opportunity to be loaded into the peptide binding groove. And when that occurs, basically CLIP just remains forever, and you sort of eventually enough time passes that the vesicle buds off with the CLIP still in the groove, and actually CLIP can be displayed on the cell surface, although this ends up basically causing the MHC class II molecule to be functionally silent. There are no TCRs that recognize CLIP as an antigen. And so those MHC class II molecules, they make it to the cell surface, but they can't activate any T cells. And so this is a process that is more immunoregulatory. HLA-DO sort of turns down the noise on exogenous antigen presentation. And so it has a role to play in the careful, tight regulation of adaptive immune responses, especially the activation of helper T cells. Okay, so these are the major steps that I wanted to cover for exogenous antigen presentation, but I'll go over one more slide which covers an important sort of caveat or exception to the rules that I've laid out for you so far, which is that endogenous proteins in the cytoplasm are processed through the endogenous pathway and are loaded onto MHC class 1 for presentation to CD8 T cells. The converse of that is that exogenous proteins are digested through the endosomal system to be loaded onto MHC class 2 to be presented to T helper cells. A major exception to that is a process known as cross-presentation. Maybe from the name you can tell that these pathways are going to get crossed somehow. So this is a process that happens uniquely in dendritic cells, and this is one reason why we think of dendritic cells as sort of the most important, the most functional, professional antigen-presenting cells, is because they uniquely have this function of breaking the rules a little bit, and they can do something called cross-presentation, which involves the presentation of exogenous antigens on MHC class 1. So remember, exogenous antigens normally are presented on MHC class 2. In dendritic cells exclusively, they can present those exogenous antigens via MHC class 1. So the way they accomplish this is they still pull in the exogenous antigens through the traditional routes, typically phagocytosis. And they degrade those antigens in the phagolysosomal system or perhaps in an endolysosome. But rather than fusing those vesicles only with vesicles containing MHC class 2, the endosomal system, the phagosomal system of dendritic cells can also express transporter molecules that actually can shuttle those exogenous peptides out of the endolysosome or phagolysosome into the cytoplasm. And this is where sort of the crossover happens. This is where you take the exogenous antigen and you feed it into the endogenous processing pathway. The way that works is that those peptides are then ubiquitinated in the cytoplasm, and when they're ubiquitinated, they can be targeted to the immunoproteasome, which does, at this point, all the things that it would normally do to a ubiquitinated protein. The protein is degraded even further. It is shuttled into the rough ER by TAP, where it can be loaded onto MHC class 1. And not pictured here is the PLC, all those chaperones, all of that same stuff is still happening. All of the same steps basically happen once these exogenous antigens enter the cytoplasm. They are basically treated from that point on as endogenous antigens, even though their origin was exogenous. So eventually, they can be trafficked back to the cell surface where those exogenous antigens end up on the cell surface, and there they can be presented to CD8 T cells. So this is helpful because, for one, CD8 T cells are very powerful effector molecules. They can kill infected cells, and they have other features beyond that also that can be protective. And this is a way that we can sort of harness the power of CD8 T cells even for extracellular pathogens. Without this process, we basically lose that entire part of our toolkit if we had a pathogen that was not intracellular. So cross-presentation by dendritic cells allows us to mount effector CD8 responses against extracellular pathogens. The other purpose of cross-presentation and why we think that it happens uniquely in dendritic cells is because dendritic cells are very motile cells. They move around the body, and they especially are good at sort of moving between tertiary tissues, meaning kind of peripheral tissues like the skin, and going into the secondary lymphoid organs like lymph nodes and the spleen in order to present antigens to naive lymphocytes, naive T cells. And so the ability to cross-present basically means that dendritic cells do not have to be actively infected to present antigen. They can just take in antigen from the environment and process it and still activate CD8 T cells that way, even if they themselves are not actively infected, meaning they don't have those pathogens in their own cytoplasm. Because frequently, if a dendritic cell is infected, its migration is impaired or it's non-functional for other reasons. It's kind of struggling because it has an infection with a bacteria or virus or whatever it is. And so cross-presentation by dendritic cells sort of overcomes multiple challenges, particularly for pathogens that are good at infecting dendritic cells. It allows us to use our uninfected dendritic cells to present their antigens. And again, it also helps us present exogenous antigens to CD8 T cells to make sure that we can sort of use our entire T cell repertoire against a broader set of potential pathogens. So summarizing this lecture on exogenous antigen processing, we saw that exogenous antigens enter the cell from the outside through endocytosis or phagocytosis, depending on the cell type. And within compartments like the endolysosome or the phagolysosome, they're broken down by acid and by proteases. And eventually, vesicles containing those digested peptides fuse with vesicles containing MHC class II. That MHC class II originates in the rough ER, but its antigen binding groove is blocked by invariant chain, which eventually is degraded into CLIP. And CLIP sort of hangs on to the last minute to make sure that the peptide binding groove is closed until MHC class II is in the presence of high affinity antigens that can be loaded onto it. We saw that antigen presentation via MHC class II is carefully regulated through various mechanisms, including the chaperones HLA-DM and HLA-DO. DM supports antigen loading onto MHC class II by chaperoning the displacement of CLIP and facilitating the loading of peptide in its place, whereas HLA-DO was a negative regulator of this process by inhibiting the association of HLA-DM with MHC class II. Finally, we saw that dendritic cells have a special function called cross-presentation, whereby they can process exogenous antigens, redirect them into the endogenous pathway, and thereby present them to CD8 T cells. And one final point here is that there's no sort of opposite form of cross-presentation. As far as we know, there is no way to take endogenous antigens and present them on MHC class II. If such a thing exists, it has yet to be discovered. It's possible, but it is not obvious, at least in terms of where the science is currently. So cross-presentation it only occurs in that direction, taking exogenous antigens and treating them as endogenous to present them to CD8 T cells. Okay, so this covers it for 2 lectures, which I said are focused on the cell biology of antigen processing. In our next 2 lectures in this module, we will shift our attention to the genetics of MHC. See you soon.